Hello, Children and Nagar. Today we're going to be solving October-November 2018 Paper 1 Variant 1. And before we start, I'd highly appreciate it if you would take a moment to subscribe and like this video. Before we start, we are offering free Topic 1 notes. All you have to do is fill in the form in the description box down below. We are also selling Topic 2, 3, and 4 notes, which include all the tips, tricks of Paper 1 and common misconceptions with worked examples too. Upon purchase, you can ask us unlimited number of questions about the topic or past paper questions for free, and you're able to exclusively contact us at any time, which is a new and unique service, and our email is in the description box down below. We also have another monthly service, which you can ask us unlimited number of questions and past paper questions for the full AS Biology syllabus only for $15 per month. Question number 1. A student made notes describing photomicrographs of 4 cells. Which table identifies the type of cell and the type of microscope used to view each photograph? Now, let's first identify the different types of microscopes, whether it's an electron microscope or a light microscope. Now, cells 1 and 2. Here we could see in the description gray cytoplasm and gray cytoplasm. If it's gray, we all know that electron microscope photomicrographs are black and white only. They do not have color. Therefore, cell 1 and 2 must be taken using an electron microscope. Now, for cell 3 and 4, here it says pale blue and green. Therefore, this is color. We could see color. And color is only available if you take this photo using a light microscope. Therefore, we know which types of microscopes this is used. Therefore, A and B are incorrect. Now, for C and D, let's read the rest of the question. Gray cytoplasm at the edge of cell contains many black lines and spots, large white area in the center. So this large white area is actually a vacuole. Therefore, this means that cell 1 is a plant cell and it's taken using an electron microscope. Therefore, this is correct. Now, for cell 2, gray cytoplasm contains many black lines and spots which fill the entire cell. Most likely, this is going to be an animal cell. Now for 3 and 4. For 3, pale blue cytoplasm surrounds a single dark blue spot. That single dark blue spot is stained using methylene blue and most likely is going to be the nucleus of an animal cell. Therefore, 3 is going to be an animal cell. This is incorrect. And for 4, many green structures are enclosed within a rectangular shape with visible boundaries. So rectangular shape within visible boundaries and green structures. These are most likely the chloroplasts. And because it says a rectangular shape, this means that the cell has a regular shape. And this is because of the cellulose cell wall determining its shape. Therefore, cell 4 must be a plant cell taken using a light microscope because it has color. Therefore, the answer must be C. Question number 2. The diagram shows a slide of transverse section of a stem. This diagram is the same as the actual size. Now, this is very important. A student observed the slide using a light microscope at a magnification of 40. The student made a plan drawing of the stem, which was 20 centimeters in diameter. So here we have the magnification and the actual size. The student labeled the plan transverse section of a stem 40. Which statement explains why this label is not correct? Now, we have to do a few calculations. Now, the actual size measured prior on paper was actually 4 millimeters. And here we have the magnification. Therefore, we could actually multiply the real image size, not the one that the student has calculated. Therefore, to do this, here we have an equation, IM, for magnification. What do is we multiply the actual size by the magnification, and the actual size from millimeters converted to centimeters is actually 40 or 0.4 multiplied by the magnification, which is 40. This gives us 16 centimeters. Therefore, this means that the calculation or the magnification is actually smaller than the one that the student drew. Now, let's see the suggestions that we have here for A. The actual size of the stem should have been checked using an eyepiece graticule. This is incorrect. You don't need to check it using an eyepiece graticule at all. It's too large to be able to do this. For B, the actual size of the stem was smaller under low power. No, it was not smaller under low power. The magnification has nothing to do with it. See, the image size in the drawing was larger than 40. So here we're talking about the image size. Here it was incorrect because we were talking about the actual size. The image size in the drawing was larger than 40 magnification. Now here, because the student labeled it as 20 centimeters and the image size that we calculated was 16, this definitely means that the image size was drawn at a larger magnification than 40, because here we have a larger diameter. Therefore, this is correct. The image size in the drawing was smaller than 40. Now, this is incorrect. Now, a very common misconception is that the students measured this area here thinking that this was the stem. However, this is totally incorrect. The area that was the stem is actually that circular area right here, because the outside or the larger circle is actually the covering slip. It is not the stem. This was a very common misconception, and many people measured the actual size of the larger one instead of the smaller one. Therefore, this is where the error came from. Therefore, the answer is going to be C. 3. Which cell structures may contain cisterna? Now, cisterna are flattened membrane vesicle found in endoplasmic reticulum and Golgi apparatus. Basically, these are each individual vesicle. These are the stacks which form the smooth endoplasmic reticulum only and the Golgi body. If you don't understand, this is what a stack of coins looks like. And each single one of those is the cisterna. Now, as we said, they are only found in endoplasmic reticulum because they're flattened stacks, and Golgi body, not in the chloroplast or mitochondrion. Therefore, the correct answer must be C. Question number 4. Which row correctly describes the function of these cell structures? Now, let's start with lysosomes, digestion of unwanted structures. This is correct. Lysosomes contain hydrolytic enzymes which carry out this function, and especially in phagocytosis, for example. So this is correct. And spherical sacs containing hydrolytic enzymes, yes, they are spherical, and there are sacs containing hydrolytic enzymes, as we just mentioned. Now, for mitochondria, abundant in sites of active transport. Now, this is not necessarily correct. Now, the reason for this is that the sites of active transport are in the cell surface membrane in channel proteins. Now, therefore, mitochondria is not available in the channel protein. And also, active transport requires ATP. So maybe ATP is what's abundant in the site, not the actual mitochondria. Therefore, this is incorrect. Also, cardiac muscle is the most abundant in mitochondria because ATP of the cardiac muscle is used in the contraction and relaxation, not in active transport. Therefore, this is also another reason why it's incorrect. Now, here for ATP synthesis, this is correct. Now, let's continue for B. Smooth endoplasmic reticulum. Now, the function of smooth endoplasmic reticulum is that they synthesize lipids and steroids. Therefore, they're responsible for lipid production. Obviously, processing proteins are incorrect and glycoproteins are incorrect too. Now, Golgi body, as we all know, it modifies and packages proteins into Golgi vesicles, which could be secreted throughout the cell surface membrane by exocytosis. Now, Golgi body, a stack of flattened stacks. This is correct, as we mentioned in the last question. Now, glycoprotein production. Yes, this is correct. As we said, they modify proteins by maybe adding a glucose unit to it. They modify and package proteins, so this is correct. And lipid production is incorrect. Therefore, the answer must be B. Number 5, the diagram shows the relationship between various cells and their components. Which row is correct? Now, let's start with A. ATS ribosomes, eukaryotic cell, mitochondrion. So now we have 1, 2, and 3. Yes, this is correct. Eukaryotic cells both have mitochondrion and ATS ribosomes in the cytoplasm. Now, 70S ribosomes, prokaryotic cells, and mitochondrion. Yes, 70S ribosomes are present inside the mitochondrion and in the cytoplasm of prokaryotic cells, so A is correct. Now for B, chloroplasts, plant cells and cell walls. Yes, plant cells do contain chloroplasts and cell walls made of cellulose, so this is correct. And for 3, 4, and 5, prokaryotic cells, they do have cell walls made of peptidoglycan or murine. But it's assuming that it has ATS ribosomes. Now this is incorrect, because prokaryotic cells actually have 70S ribosomes. Therefore, B is incorrect. For C, circular DNA nucleus and eukaryotic cells. Here it's assuming that the nucleus has circular DNA. Now this is totally incorrect because the nucleus has a normal linear DNA made of 2 chromatids. Therefore, C is also incorrect for D, prokaryotic cell circular DNA in chloroplast. Now here, prokaryotic cells do have circular DNA in the cytoplasm, so this is correct, and chloroplasts also do have circular DNA, so this is correct. Now, membrane bound here, chloroplasts are membrane bound, so this is correct by double membrane. However, here's assuming that ribosomes are membrane bound. However, ribosomes are actually not covered by membrane whatsoever. They are non-membrane bound. Therefore, this is incorrect, and the correct answer must be A. Number 6. Which comparison of bacterial cell walls and plant cell walls is correct? Now let's start with bacterial cell walls made of polymer of alpha or beta glucose. Now this is incorrect. Bacterial cell walls are made of murine or peptidoglycan, which are actually proteins 100%. They do not include any glucose units. Therefore, all these are incorrect, and peptidoglycan is correct because it's made of protein. Now, plant cell walls made of cellulose. This is correct, cellulose by the condensation of beta-glucose monomers. For B, made of a polymer of amino sugars, amino is incorrect. Made of polymer of alpha glucose. Now, alpha glucose is incorrect. We said cellulose is made of 100% pure, only beta-glucose monomers. Therefore, this is correct, and the correct answer must be D. Question number 7. A glycosidic bond is broken and 2 monosaccharides are formed during a positive test for a non-reducing sugar. Now, this is very important. Which row identifies the catalysts and reactants in this process? Now, let's first divide the question and start with breaking the glycosidic bonds. Now, breaking the glycosidic bonds is a hydrolysis reaction. In a hydrolysis reaction, you actually add water at high temperatures. Therefore, the catalyst for this hydrolysis reaction must be hydrochloric acid, and this breaks the glycosidic bonds, not sucrase enzyme. Now, the reactants here, it's saying it forms a positive test for non-reducing sugars. Therefore, the reactants must be non-reducing sugars. In this case, A is incorrect because fructose is actually a reducing sugar, same with glucose. Therefore, the correct answer is sucrose, and water has nothing to do with it. Therefore, the answer is going to be B. Number 8. Which pair of statements are correct? A. Carbon and oxygen occur in a ratio of 2 to 1 in carbohydrates. Now let's take glucose as an example, C6H12 and O6. Therefore, as you can see here, they occur in 1 to 1 ratio. Therefore, they're in 1 to 1, and this is incorrect. Now, triglycerides are soluble in water. They're insoluble. This is incorrect. Why? Because they are hydrophobic. They might be soluble in ethanol, for example. Now for B, glycolipids are found in all cell surface membranes. This is correct because glycolipids act as cell recognition molecules as antigens. Therefore, all cell membranes, to make the cell distinguishable, must have glycolipids or antigens on them. Therefore, this is correct. Now, carbohydrates are stored as starch in plants. Yes, starch is the storage polysaccharide found in all plants. Now for C, phospholipids all have 2 saturated hydrocarbon chains. Now, the trick is with the wording of this question. Phospholipids do have 2 hydrocarbon chains, but not necessarily saturated. They could also be unsaturated. Therefore, this is incorrect. And polysaccharides are polymers. Yes, this is correct. Polysaccharides are sugar monomers put together, making a polymer. So therefore, this is correct. D. Water is released during the formation of a glycosidic bond. So during formation of a glycosidic bond, this is a condensation reaction. And water is formed in a condensation reaction. Therefore, this is correct. And phospholipids all have 3 ester bonds. Now, this is incorrect. The reason for this is that phospholipids are made of a glycerol molecule bound to 2 fatty acid chains by 2 ester bonds only, because the third here we have inorganic phosphate, which is not bound to an ester bond. Therefore, it only has 2 ester bonds, and this is going to be incorrect, and the answer must be B. Question number 9. What is the general formula of cellulose? Now, we have a trick used to calculate any polysaccharide. Now, to do this is that we know that the formula for glucose is going to be C6H12O6. Now, we know that forming a cellulose is formed by a condensation reaction, and in condensation reactions, water are released. So by the condensation reaction of beta-glucose monomers. Therefore, because we know that a molecule of water is formed, and here it's N, therefore what we could do in case of polysaccharides only is we subtract the atoms from the glucose monomer. In this case, it's going to be C6H10 because we subtracted 2 hydrogen atoms, O5. Therefore, the correct answer in this case is going to be C. Question number 10. Triglycerides consist of glycerol and 3 different fatty acids: linoleic acid, oleic acid, and palmitic acid. The diagram shows one possible arrangement of the fatty acids LOP in the molecule. What is the total number of different arrangements of the fatty acids in this triglyceride? Now, during calculations, it's only going to be 3 different arrangements, including this one, and I'm going to show you why. Now, for the second arrangement, we could have OLP. And for the third unique arrangement, we could have LPO. We can only have those 3. The reason for this is that if we try to do, for example, POL, and we all know that triglycerides molecule could actually be rotated 180 degrees. If we rotated that, we would end up with LOP, so it's exactly the same triglycerides. Therefore, it's not a different arrangement. Same with OLP. If we try, for example, to arrange PLO, we know that triglycerides could rotate, so this would also give us OLP. Therefore, this is also not unique, and vice versa for this one too. Therefore, we could only have those 3 different arrangements, and the answer is going to be A. Question number 11. Which statements about a peptide bond are correct? 1. It joins 2 monomers which always identical to each other. This is incorrect. They don't necessarily have to be identical, and that's what gives the primary structure its unique structure. Now for 2. It contains 4 different atoms. Yes, the peptide bonds do contain 4 different atoms. Let's draw them. Therefore, this is correct. 3. It can be broken by addition of water at room temperature. Now, peptide bonds could be broken by the addition of water in a hydrolysis reaction. This could potentially break the peptide bond. However, here it says at room temperature. We all know that peptide bond is actually a very strong covalent bond, therefore requires a very large amount of energy by addition of water to break the peptide bond. Therefore, room temperature is never enough to break the peptide bond. Therefore, this is incorrect. For 4, it is important in the primary structure of proteins. Yes, peptide bond is what actually binds the different arrangements of amino acids, forming the primary structure. Therefore, this is correct, and the correct answer is 2 and 4, which is D. Statements about the primary structure of a protein are correct. 1. It may be branched. This is incorrect because the definition of primary structure in the first place is a linear sequence of amino acids. A linear sequence. It could never be branched. Therefore, this is incorrect too. Is determined by the sequence of DNA bases. Yes, this is correct because the codons on the DNA is what actually forms the amino acids by transcription and translation in the ribosomes. This is correct. 3. It is unique to that protein. Yes, every protein has a different arrangement of amino acids. And we all know that amino acids have different R groups, forming different properties. Therefore, it is unique to every single protein. This is correct. 4. It determines the tertiary structure of a protein. Yes, this is correct because I told you prior that each different amino acid has a different R group, and every R group has a unique tertiary structure bond precisely formed to it. Therefore, yes, it does determine the tertiary structure, and a change in a single amino acid molecule could completely alter the tertiary structure. For example, valine in sickle cell anemia. This could totally alter the tertiary structure. Therefore, 2, 3, 4 is correct, and the answer is going to be D. Mutation occurred within the DNA sequence coding for an enzyme, causing a decrease in the rate of a reaction catalyzed by this enzyme. Which statement could explain the decrease in the rate of reaction? Now 1. An inhibitor for this enzyme has an increased affinity for the enzyme and forms an enzyme inhibitor complex more easily. Now, when a mutation happens within the DNA, this means that the tertiary structure changes. This also means that maybe the tertiary structure has changed to a shape which is more complementary and has a higher affinity to the inhibitor instead of the substrate. It might be more complementary to the inhibitor. Therefore, it has a higher affinity to it. Therefore, this is a valid point. This is correct. For 2, the active site of the enzyme might have changed shape and is no longer complementary. So here the trick with the wording is no longer complementary. Here in the question it says causing a decrease in the rate of reaction. If it was no longer complementary, as it is mentioned here, then the reaction would totally stop because no substrate could be longer converted to product. However, in this case, it says causing a decrease, not completely stopping the reaction. Therefore, no longer complementary is incorrect. It might be partially incomplementary, but no longer complementary at all is incorrect. 3. The activation energy for the reaction with the mutated enzyme is greater than with the non-mutated enzyme. Of course, this would lower the rate of the reaction. The reason for this is that activation energy increased for the second one. This means that more energy is required for the reaction. If more energy is required for the reaction, this means it's going to have a lower rate than the original one with lower activation energy. Therefore, this is also a valid point, and the answer is going to be C. Question number 14, which row is correct for an enzyme with a low Michaelis-Menten constant? So Michaelis-Menten constant measures the affinity for the enzyme for the substrate. Therefore, a low Michaelis-Menten constant equals a higher affinity, and vice versa. They are inversely proportional. Now, affinity of enzyme for substrate, as we said, because it has a low Michaelis-Menten constant, this means that it has a higher affinity, and substrate concentration at maximum reaction rate. So we know at maximum reaction rate, most of the substrate is actually converted to products. This means that the substrate concentration at max reaction rate is actually going to be the lowest, because most of it is converted to product in this case. Therefore, the correct answer must be B. Question number 15, the diagram shows a glycoprotein embedded in the cell surface membrane of a human red blood cell. This glycoprotein is part of a system of cell surface blood groups recognition sites. Which row identifies the role of this glycoprotein and regions P and Q of this molecule? So here it says part of a system of recognition sites. Recognition sites clearly means that this is going to be an antigen and might also be a receptor, but never a carrier or a channel. Now, we have here different regions, P and Q. Region P is at the core of the cell surface membrane, and the core of the cell surface membrane is hydrophobic. This means that the region P must also have amino acids which are hydrophobic. Now for Q, because it's at the cytosol or forms hydrogen bonds with the interior fluid of the cell, this means that it must be hydrophilic. It must have amino acids which are actually hydrophilic. Now let's see the suggestions that we have here. Here we have amino acids hydrophobic in P. This is correct. Amino acids hydrophilic are groups. This is incorrect. Same with here. This is also incorrect. And this is correct. Therefore, the correct answer in this case must be A. Question number 16. The diagram shows the transport of ions across the cell surface membrane. Inside the cell, there's a low concentration of sodium ions and a high concentration of potassium ions. So the most important thing in this question is to actually label the diagram. So inside the cell, there's a low concentration of sodium ions and a high concentration of potassium ions. And outside the cell, which is here, there's a low concentration of potassium ions. Let's draw this here, and a high concentration of sodium ions. Now, which ionic movements are represented by the arrows? So here we have diffusion through the core, because diffusion does not need a carrier molecule, and active transport. Active transport is definitely going to be within that carrier molecule because it requires a carrier molecule and ATP. Therefore, in active transport here, we're looking only at 2 and 3. Now, here we could see that inside the cell, there's a lower concentration of sodium and a higher concentration of sodium outside. And we all know that the definition of active transport is the movement of ions against concentration gradient. Therefore, sodium must have moved by active transport across at 3. Therefore, sodium must be 3. Sodium ions must be 3. Same with potassium, because active transport, the ions move against concentration gradient. Here for potassium, outside the cell, we have a low concentration of potassium, and inside we have a higher. Therefore, it moves against concentration gradient. therefore means that potassium is going to be 2. Now for diffusion, we're only looking at 1 and 4 because, as we said, diffusion could happen through the hydrophobic core and through the cell surface membranes. They do not require a carrier molecule. Now, the definition for diffusion is that it happens down concentration gradient from higher concentration to lower. Therefore, let's start with the sodium. Outside, we have a higher concentration, therefore it diffuses to the lower down concentration gradient. Therefore, sodium must be 1. Same with potassium. Inside there is a higher concentration, therefore it must diffuse down concentration gradient. Therefore, potassium is going to be 4 and the answer must be A. Question number 17. The indicator cresol red changes from red to yellow when put into an acid. Some blocks of agar containing cresol red were cut to different sizes and put in an acid. All other variables were kept constant. The blocks were measured in millimeters. Which block became completely yellow most quickly? So the block which became completely yellow most quickly has definitely a higher surface area to volume ratio. Why? Because diffusion happened the fastest. And factors that increase diffusion is a higher surface area to volume ratio. Now, let's here look at the value which has the higher surface area to volume ratio. So, in this question, where there are variable numbers, I prefer using common sense instead of calculating every single calculation because this is very time consuming during the actual exam. Therefore, for A, for example, if we would draw it by common sense, we all know that 30 will have a huge surface area, and for 3 it would look like this, for example. Therefore, there's a much higher surface area to volume ratio for A. However, compared to B, this is a cube because they're all the same dimensions, so it would look something like this. And a cube definitely does not have a higher surface area to volume ratio than a surface like this, for example. Same thing with C here. If we would draw it, it's smaller than 30. This means it would look something more like this, because of the 6 here. But it seems like A has a higher surface area to volume ratio. Now, same thing with D. Here it's a cube. And as we know, a cube actually has the lowest surface area to volume ratio. Therefore, by common sense, A must be the correct answer. 18. When red blood cells are put into pure water, they burst by hemolysis. Which statement explains this hemolysis? Now, when red blood cells are put into pure water definitely has a higher water potential than the inside. The inside, because of these solutes and particles present, it has a lower water potential. And as we all know, by osmosis, water goes from a higher water potential to a lower water potential. And what happens is the cell keeps swelling until it bursts. Now, the reason why it bursts is because it only has a cell surface membrane. It doesn't have a cell wall, for example, like a plant cell, that would actually withstand that pressure. This means that it just keeps swelling and filling up with water until it bursts. Now, let's see the statements that we have here. 1. The water potential of the surrounding liquid is lower. Now, this is incorrect because the water potential of pure water is definitely higher. For 2, the cell surface membranes of red blood cells are not supported by cell walls. Yes, this is the reason why it bursts. 3, more water moves into the red blood cells by osmosis than leaves these cells. Yes, by osmosis, water moves by net movement. Net movement means that yes, water is moving from a higher water potential to a lower water potential, but in a greater quantity to what's leaving. Okay, so a greater quantity, but there is still a net movement. So water is entering at a larger rate, and there's also a little bit of water leaving. Therefore, this means that this is correct. For 4, water enters the red blood cell by osmosis but does not leave the red cells. Does not leave the cells. Now, this is incorrect because the definition of osmosis is net movement. So net movement, as I just mentioned here, water would enter in at a greater quantity, but still some water would also leave. Therefore, this means that it does not leave the cells. This is incorrect, and the correct answer is 2 and 3, which is C. Question number 19. How many copies of each DNA molecule will be found in a cell at each stage of mitotic cell cycle? Now, we all know that after mitosis, we are left off with 1 copy of the DNA molecule. And S phase, so in G1, we have 1 copy. And in S phase is the DNA synthesis phase. That copy is duplicated. Therefore, in S phase and anything that comes after the S phase, G2 is also duplicated and has 2 copies of DNA up until the end of cytokinesis. So G1 only has 1 copy of that DNA molecule. Now, cytokinesis, yes, this is the stage that the cell starts dividing. Therefore, it makes sense that it would only have 1 copy of DNA. However, this is incorrect because it does not specify whether it's the end of cytokinesis or the beginning of cytokinesis. Cytokinesis, the cell looks like that. It hasn't yet divided. Therefore, it still has 2 copies of the DNA because it's cell at each cell, not at each nucleus. Therefore, it still has the same 2 copies that were duplicated during the S phase at DNA synthesis. Therefore, this is correct and the answer must be B. Which metabolic process will be very active in a cell that has just completed cytokinesis? So, in a cell that has completed cytokinesis, it's small. So it actually needs to grow, and this takes place, by the way, in G1 phase, after it has completed cytokinesis. So to be able to actually grow, it needs to synthesize proteins. Therefore, ATP formation is essential in protein synthesis. Therefore, this is correct. For 2, DNA replication. In G1 phase, we do not have to need anything to do with DNA replication. The reason for this is that DNA replication actually takes place in the S phase, which is after the G1. Therefore, this is incorrect. 3, protein synthesis, as we mentioned, this is what actually helps this cell to start growing. Therefore, this is correct, and the correct answer must be 1 of 3, which is B. Question number 21, a mutation occurs in a gene which prevents the production of telomerase. What is the effect of this mutation if it occurs in bone marrow stem cells? So, telomeres are sequence of DNA found at the end of chromatids. So, these prevent the loss of genes and prevents gene shortening when the cell starts to divide, because each time the cell divides, it loses parts of its telomeres. And telomeres is what determines the age of the cell, because if the full telomeres have been removed from the end of the chromatids, this means that the DNA by cell division is going to start to get lost. And if the DNA starts to get lost and starts to shorten, this means that the cell most likely won't make it. And therefore, telomerase enzyme is what actually synthesizes the telomeres to keep the stem cells dividing over and over again without losing any DNA. Therefore, what is the effect of the mutation? Let's look at the suggestions that we have here. A rapid increase in the production of lymphocytes. So as we all know that bone marrow cells forms the white blood cells, and if the production of telomeres decreases, there will actually be a decrease in lymphocytes, because I told you the bone marrow cells can no longer divide to produce cells which could actually differentiate. So this is incorrect. B, tumor grows in the bone marrow. So this is incorrect. Telomeres have no relationship with tumor growth. Tumor grows due to mutations or carcinogens. Therefore, this is incorrect. C, bone marrow stem cells eventually no longer divide. Yes, this is correct, because I told you, if the telomeres have gotten to a point which all of it is removed and it's not restored by telomerase, this means that the bone marrow stem cells will start to lose DNA, and if it loses DNA, then it can no longer divide and it will just die. Therefore, C is going to be the correct answer. D, the total blood cell count will be unchanged. This is incorrect. It will actually decrease. Number 22. What are the products when DNA molecule replicates? A, 2 molecules of DNA, each made of a paired sequence of bases. So here, when we're describing the DNA, we actually use the proper terms. For example, in this case, we use molecules to describe DNA, not strands, and we also use the monomers that make up the DNA when describing it. So the monomers for DNA are actually not the bases. Bases are 3 nucleotides. The monomers for DNA are nucleotides. Therefore, we're using nucleotides, and we use 2 molecules of DNA, not strands. Therefore, the answer is going to be B. Question number 23. The following translation, the alpha polypeptide chain of hemoglobin alpha globin undergoes modification. During this modification, the first amino acid is removed, leaving 141 amino acid residues. How many nucleotides does the gene coding for alpha globin contain? So first of all, we have to find the total number of amino acids before the modification to be able to find how many nucleotides. Now here it says the first amino acid is removed. So the total amino acids originally were 142. Now, as we all know, 3 nucleotides or a codon, so 3 nucleotides or a codon, codes for 1 amino acid. Therefore, to calculate how many nucleotides the gene containing for alpha globin, we must multiply 142 by 3 to get the number of nucleotides. 142 is the number of amino acids and 3 is the number of nucleotides forming it. Therefore, the correct answer must be D. Question number 24. Which statements correctly describe transport pathways in dicotyledonous plants? Now, 1. In the apoplast pathway, water does not move through the plasmodesmata. So let's clarify a few points. In the apoplast pathway, this is the intracellular pathway. For example, water only moves through cell walls. They never ever enter a cell in the apoplastic pathway. Now, in the symplastic pathway, water actually goes into the cell through the plasmodesmata by osmosis. So they actually cross the surface membrane and enter the cell. So let's see suggestions that we have here. In the apoplast pathway, water does not move through the plasmodesmata. Now this is correct because, as we said, the apoplastic pathway is the intracellular pathway and it only moves through the cell walls. So they never enter the cell. 2. The symplast pathway, water does not move through intercellular spaces. Now this is correct because we said that water actually diffuses into the cell by osmosis. 3. The apoplast pathway may be blocked by the Casparian strip. Now this is correct. The Casparian strip actually blocks the apoplastic pathway. The reason for this is the Casparian strip is actually made of a waterproof material called suberin. Suberin does no longer allow the apoplastic pathway to take place. It only allows the movement inside by the symplastic pathway. Therefore, all 3 is correct and the answer must be A. Question number 25. The photomicrograph shows a transverse section through a root. What is the simplest ratio of the diameter of the root? Now, here, first of all, the first thing to note in a ratio is that it never has units. And by the way, we do not actually need to measure anything in this situation. So we all know that this is going to be incorrect because ratios don't have units. And here, by common sense, the simplest ratio, we have 40:11 and 80:22. The simplest ratio must be 40:11 because here it could be simplified further. Therefore, we do not actually need to do any calculations and the answer must be B. Question number 26. Which properties of water molecules are important in the upward flow of water through the xylem? So the most important properties are actually adhesion and cohesion. So adhesion is the attraction between the water molecules and the cellulose cell wall due to the hydrophilic properties of cellulose. So this is the attraction between the water molecules and the cellulose cell wall of the xylem. However, cohesion is the hydrogen bonding between water molecules. Because they are able to form hydrogen bonds between each other. And both of these qualities actually helps the water go upwards by mass flow. Now let's see the suggestions that we have here. 1. Water molecules are attracted to each other by hydrogen bonding. Now this is correct in cohesion. 2. Water molecules are attracted to cellulose by adhesion. Yes, this is correct due to cellulose hydrophilic properties. 3. Water molecules have a high cohesion in water columns. Yes, this is also correct. Therefore, the answer must be A. Number 27. What's correct for a phloem sieve tube element that is unloading to a sink? So let's draw a diagram to make it more clear. So let's assume that this is the sink cell and phloem or the sucrose is unloading into the sink cell. So now what's going to happen, because actually sucrose is moving from the phloem sieve tube elements into the sink cell, this means that the sucrose concentration in the phloem sieve tube elements is going to decrease. Therefore, the water potential inside the phloem sieve tube element will actually increase. Therefore, the water potential would be less negative. Less negative here means a higher water potential than sink. Now, lignification of the cell wall. So, cell walls of phloem sieve tube elements are not actually lignified. What is lignified is the xylem vessel elements. Therefore, lignification in phloem sieve tube elements is actually going to be absent, and the answer must be A. Question number 28. Which statement concerning transpiration is correct? So transpiration is loss of water vapor through the stomata. And factors which increase the rate of transpiration is increasing the water potential gradient and increasing the airflow and increasing the temperature. Now let's see suggestions that we have here. A. On a humid day, the water potential gradient between the intercellular airspace and the external environment increases. Now this is incorrect. The water potential actually decreases. Why, you would ask? Because in a humid day, the concentration of water particles in the atmosphere decreases. Therefore, this lowers the water potential gradient and therefore lowers the rate of transpiration. So this is incorrect. B. Water arriving at the spongy mesophyll cells via the symplast pathway must move by osmosis through the cell surface membrane before evaporation from the surface. Yes, because symplastic pathway is the movement of water through the plasmodesmata into the cell through the vacuole by osmosis. Therefore, in the symplast pathway, it must move by osmosis through the cell before it evaporates. Therefore, this is definitely correct. First, C. Water diffuses down the water potential gradient from the saturated airspace through the guard cells before evaporating. Now the order and the sequences here are actually incorrect. It evaporates first, then it diffuses through the stomata to the atmosphere. Therefore, this is incorrect. D. Water moves up the xylem in the apoplast pathway and can continue on this pathway by osmosis. Now, apoplastic pathway is the intracellular movement of water through cell walls only. In apoplastic pathway, water never ever enters the actual cell. Therefore, it does not enter by osmosis at all. Therefore, this is incorrect, and the correct answer must be B. The section through the heart rate and associated blood vessels. What is correct for the flow of blood through the heart? Now, first of all, we must start by labeling the heart. Now, in order to do this, we have to look at the most muscular side. The most muscular side is going to be the left side. The reason for this is that the left side must pump blood to the rest of the body, so needs increased cardiac muscle in order to generate enough force to push the blood to the rest of the body. Contrasting the right side only needs to push the blood to the lungs, therefore has low resistance. Therefore, we first start by labeling the heart. We all know that this is going to be the left side, and this is going to be the right side because there's more cardiac muscle in this side. Now, we all know that this is going to be the atria, 2 is going to be the atria, and or 3 is going to be the right atria, and 4, because it's on the right side, then this must be the pulmonary artery, because this is coming out of the ventricles and leads to the pulmonary artery and leads to the lungs. Now for 2, because it's coming out of the left ventricle, is definitely going to be the aorta, which pumps blood to the rest of the body. Now, what is the correct sequence of blood flow? Now let's look at the suggestions that we have here for A. 1, 2, 3, and 4. So 1 is the aorta, 2 is the atria. Now, it's impossible that there's backflow from the aorta back to the atria because blood, first of all, atrial systole takes place, the contraction of atria, then blood goes to the ventricles, then out to the aorta. So the order here is incorrect. For B, 2, 1, 3, and 4. So in this case, 2 and goes to 1, so 2, then atrial systole goes to the ventricle, then ventricular systole takes place, and blood is pushed through the aorta. Therefore, this is correct. Then, for 3, it comes back to the vena cava, so this part here is the vena cava, to the right atrium. And this is deoxygenated blood, so this is also correct. And for 4, then the blood here, atrial systole takes place, blood goes to the ventricle, ventricular systole takes place, and blood goes out to the pulmonary artery, deoxygenated blood, to go to the lungs to get oxygenated. Therefore, this order seems to be correct, and the answer must be B. Question number 30. Blood, tissue fluid, and lymph each have a different composition. Which row shows the composition of lymph? So lymph, this is a general rule you all must memorize. It's obviously made of water and it contains antibodies because it contains white blood cells. So this is correct. And lymph contains lipids because in the small intestine, lipids are actually absorbed through the lymph. Therefore, this is correct and the answer must be A. 31. Which row shows the change in concentration of some substances in red blood cells when carbon dioxide diffuses from active cells? Now, here let's start with carbonic anhydrase. So carbonic anhydrase is the enzyme found in red blood cells. So even if the carbon dioxide concentration does increase, the enzyme concentration in the red blood cell is not altered in any shape or form. Therefore, this is correct. There's no change. Now here we have hydrogen carbonate ions and hydrogen ions. Now when the carbon dioxide concentration increases, what happens is that more carbon dioxide now combines with water molecules to form carbonic acid by the action of carbonic anhydrase. To form carbonic acid by the action of the enzyme carbonic anhydrase, which we said is not changed. Then after carbonic acid has formed, obviously this is when carbon dioxide concentration increases, it dissociates into hydrogen ions and bicarbonate ions. Therefore, if the concentration of carbon dioxide increases, then the concentration of both hydrogen ions and bicarbonate ions also increases. Therefore, the answer must be D. Question number 32. The diagram shows the pressure changes in various structures of the left side of the heart during the cardiac cycle, at the end of which period is the ventricle full of blood. So here we're looking for ventricular diastole. This means that it's full of blood and it's not in a contraction phase. So the ventricle is not contracting in that phase. Now, let's break up the diagram. Here, this is the line for the left ventricle and the pressures. Now, when the pressure of the left ventricle increases, for example here, this means that is ventricle systole taking place. So ventricular systole is when the ventricle starts to contract. Obviously, when the ventricle starts to contract, this means that it has a lower volume of blood. Therefore, B and C is incorrect. So when the pressure increases, this means that the ventricle is contracting, therefore it has a lower volume of blood. Now here it says at the end of which period is the ventricle full of blood. Now, right before ventricular systole, there's ventricular diastole. So at the end of A, at a very low pressure, here the ventricle is full of blood. So A is going to be the correct answer. Here D is incorrect because here it says at the end of which period is the ventricle full of blood. However, it's only at the beginning where the ventricle is actually still filling with blood and it's not yet filling with blood. It says here at the end of which period. It's still not full of blood. Therefore, the correct answer must be A. 3. The photomicrograph shows a cross-section through the lining of part of the respiratory system. Which statements are correct? So, 1. Goblet cells are visible between squamous epithelium cells. Now, in this case, we cannot actually see goblet cells because goblet cells do not have cilia. But here, all the cells that we could see contain cilia. And by the way, this is not a goblet cell, this is mucus. Therefore, this is actually incorrect. There are no goblet cells and they are not visible. 2. Smooth muscle is visible. Yes, smooth muscle, we could see it right here underneath the cartilage. Therefore, it is visible. 3. The section cannot be from a bronchiole as cartilage is visible. Yes, this is cartilage. Therefore, the answer must be 2 and 3, which is D. Question number 34. In the lungs, oxygen and carbon dioxide pass through the cell surface membranes by diffusion. Which row is correct? And here it's asking for the number of cell surface membranes diffused through by oxygen and carbon dioxide. Now let's first just draw a diagram of the blood capillaries and the alveoli. Now, for the oxygen to diffuse from the air into the alveoli, then the endothelial lining of the blood vessel, then into the red blood cell, let's count how many cell surface membranes it will actually pass by. Now, first of all, we know that the alveoli is made of a layer of simple squamous epithelium. And squamous cells, this is what they look like. They are flat cells and they look almost like a fried egg. So here, the alveoli is covered by squamous epithelium cells, by simple squamous. Therefore, in order for oxygen to actually pass just through the alveoli, it needs to first enter the squamous epithelium cell, so that's the first cell surface membrane. Then, in order to exit the squamous epithelium cell, it has to pass through the next cell surface membrane. So, first to enter and to exit. Therefore, in the alveoli, it needs to pass through two cell surface membranes. Now, through the endothelial lining of the blood capillaries, it is also made of a layer of the simple squamous epithelium, so that same cell that we have here. So therefore, in order to also enter the simple squamous and exit it, it has to enter through other two cell surface membranes. So here we have two, then in the blood capillaries we also have two. Then, in order to enter the red blood cells, the red blood cell, we all know it has only a single membrane at the outside. So it has to pass through another cell surface membrane. Makes a total, if we count, 2 in the alveoli, 2 in the endothelial lining of the blood capillary, and 1 in the red blood cells. That's a total of 5. Therefore, this is correct. Now, carbon dioxide to air. Now, there are two locations for carbon dioxide. There's in the cytoplasm and there's inside the red blood cell as carbaminohemoglobin. Therefore, let's assume that now we're looking from the diffusion of the plasma to the alveoli. Now, as we mentioned here, it has to diffuse back through the endothelial lining, which is made of a layer of simple squamous epithelium. So that's here 2 membranes here, and to pass out of the alveoli, it also needs 2 membranes. So in case if it's in the plasma, it needs to pass by 4 membranes. But in case if it's inside red blood cell, it needs to pass first outside the membrane. So that's the first membrane here, then to the endothelial lining, and then to the alveoli outside the lungs. Therefore, in the case which is inside red blood cell, here it's going to pass through only 5. Therefore, here it could be 4 or 5. This is correct, and the answer must be D. Question number 35. Which flow diagram correctly describes the effects of tar entering the lungs? So A. Carcinogens come into contact with DNA. First of all, let's clarify a point that tar is a carcinogen which basically forms tumors at the long-term use of smoking. Now, carcinogens, mutation occurs, uncontrolled cell division. Yes, carcinogens cause mutations, therefore cause cancers and uncontrolled cell division, so that's correct, and the correct answer is A. For B, goblet cells secrete more mucus. Mucus accumulates, causing infection. Now, this is incorrect wording. The reason for this is more mucus is not what causes the infection and the accumulation of phagocytes. What actually causes the infection is pathogens, not the increased number of mucus. Therefore, this is incorrect. Same here. The same concept. This is incorrect. D. Mutation occurs, phagocytes attracted by inflammation. Now, mutation has nothing to do with phagocytes. Phagocytes would accumulate in case of an infection, but mutations or cancers are non-infectious. Therefore, this is incorrect, and the answer must be A. Number 36. Which list contains only infectious diseases? So, cholera, HIV, lung cancer is non-infectious. This is incorrect. B. Cholera, malaria, tuberculosis, and sickle cell anemia. Sickle cell is genetic, where CTT is replaced with the CAT codons. Therefore, this is incorrect. C. HIV, AIDS, malaria, measles, and tuberculosis. Yes, HIV is a virus, malaria is a eukaryotic infection, measles is a virus, and tuberculosis is a bacterial infection. Therefore, the correct answer must be C. And which disease is caused by a eukaryote? So, a disease used by a eukaryote is definitely going to be malaria, and its vector is going to be female Anopheles mosquitoes. So, malaria is the correct answer. Number 38. A vaccine is available against most of the common strains of influenza virus. Which statement explains why the vaccinated people are not immune to a mutated influenza virus? So, when a vaccine is introduced to the body, it has unique antigens only unique to a specific influenza virus. Therefore, memory cells are only produced unique to the antigen presented in the vaccination. So in case that the influenza mutated, this means that the antigens have changed. Therefore, there are no longer memory cells complementary to that specific antigens. So, therefore, this means that they are not immune because memory cells are no longer present. Now let's see the suggestions that we have here. 1. They will not have primary immune response to the mutated antigen. Now, this was a very common misconception. This is wrong. They will still have their same immune response. This is correct, but not having a primary immune response is incorrect. So 2, they will not have a secondary immune response to the mutated antigen. So in this case, this is correct. They will not have a secondary, they will have the primary. Well, the body starts forming new memory cells to the new antigen that has been demonstrated in a normal immune response. Now, they will not have a secondary immune response. Why? It's simply because there are no memory cells present for this unique antigen. Now, number 3, their memory cells do not recognize the mutated antigens. Now, this is correct, we just mentioned, because the memory cells are only complementary to a specific one, not to the mutated new one. Therefore, the correct answer must be D. Question number 39, which statements correctly describe lymphocytes? So 1, each B lymphocyte has the ability to make several types of antibody. This is incorrect. Each lymphocyte has cell receptors only complementary to a specific antigen only. Therefore, it only produces a specific antibody only for that complementary antigen. Therefore, this is incorrect. 2, some B lymphocytes and T lymphocytes become memory cells. Yes, there are memory B and memory T lymphocytes for a complementary antigen. 3, plasma cells secrete antibodies into the blood plasma. Yes, plasma cells do secrete antibodies. 4, some T lymphocytes stimulate macrophages to kill infected cells. So, some T lymphocytes, which is the T helper one, so be careful, it's the T helper, not the cytotoxic one. So the T helper secretes cytokines which activate macrophage to be more aggressive and kill infected cells by phagocytosis. So it's the cytokines secreted by the T helper which activates the macrophage. So 2, 3, and 4 is correct, and the answer is going to be also D. Question number 40, Addison's disease can occur when antibodies are produced in response to an enzyme found in some organs of the body. Which statements correctly describe Addison's disease? 1, it's a non-infectious disease. So first of all, Addison's disease is an autoimmune condition where antibodies are produced against the adrenal glands found on the top of the kidneys. Therefore, the body cannot produce enough adrenaline. Therefore, let's start with 1, it is a non-infectious disease. Yes, it's an autoimmune condition. 2, it's a type of autoimmune. Yes, this is correct, where antibodies are produced against self-antigens. So this is also correct. Therefore, the correct answer must be A. Thank you very much for watching this video. If you found it helpful or it offered you any value, I'd highly appreciate it if you would subscribe and like this video. And if you have any past paper recommendations, please leave them in the description box down below. Thank you very much.